On this episode of Sea Science, crew members from the research vessel Melville gear up for a 9-day trip to California's Santa Barbara Basin.
Is our home away from home?
As equipment and supplies arrive on the docks in San Diego, scientists from the Scripps Institution of Oceanography prepare for their own journey.
It's only a matter for maximizing our use of the stratigraphy inside the box core.
For the scientists on board the RV Melville, this journey will be an exploration into the past, a mission to explore the deep ocean basins of Southern California. Using a unique set of tools, both large and small, scientists will analyze the ocean's water and take sediment samples from the last glacial period.
The Santa Barbara Basin was a site chosen for our studies because it is an anoxic basin, which means that there's very little oxygen at the bottom. And because of this, there aren't many animals that are burrowing and churning up the sediment. And therefore, the sediment actually layers on top of itself with light layers during the summertime and dark layers at winter. And so this almost creates a tree ring effect. And so as scientists, when we core down into the ground, we're able to count back the rings or layers back into time and sample particular periods of interest. So in Santa Barbara is where they were able to put together some of the first climate records of what the weather and climate patterns have looked like in the past. And we've also been able to see a lot of changes in the animals and productivity and chemistry of the waters, as well as changes that have taken place on land, like movements of trees and different plants. And for these reasons, it's a great place to be able to go and see what's happening today and then see what has happened in the past.
Aboard the RV Melville, crew members prepare the ship for voyage, and that means safety checks. The crew starts with a man overboard drill. As scientists put on the life suits, the ship staff makes a few safety announcements.
Before you get in and bring them in with you, like I said, you guys are all going to have to do this too, so it only lasts so much. Thank you, Chris. Don't give me applause.
I think if you just stick it in a cold refrigerator.
Research vessels like Melville are floating labs equipped with both navigational and scientific equipment. The scientists have already loaded the largest equipment on board the ship, but they still need to set up their labs with microscopes and computers.
They're quite common.
To the west, where we don't have an anoxic bottom anymore, we should find a lot of interesting animals sitting on the surface, which is always fun to go and see the brittle stars and sea cucumbers and things like that.
As the ship heads for the Santa Barbara Basin, the scientists head for their
This
quarters.
is the hallway into the living quarters.
Basically a bed,
We're obviously-
some drawers, and maybe your own bathroom.
9 days with Christian. He's a graduate student from SIO. I grabbed the bottom bunk because he's smaller, so he's going to be in the top.
This is for someone's project with the Humboldt squid, and so we're going to be throwing these glowing lures over the side, and hopefully they'll just lash onto it and we can pull them up.
After setting up their labs and berthing areas, the scientists and ship's crew prepare for 12-hour shifts, 24 hours a day for the next 9 days. The Melville will travel overnight 300 miles north to the Santa Barbara Basin, allowing the crew to prepare for their first experiment at 6:30 a.m. The plan is to launch the piston core, a high-performance coring device that will sample sediment as old as 18,000 years. Together with the box core, which samples the top layer of sediment down to 200 years, and the casting core, which samples down to about 2,000 years, the piston core is a monster. Comprised of multiple sections to create a 20-meter long sampling tube.
One of the devices we have for collecting a very long sediment core, which might be between 15 and 18 meters in length, and therefore capable of giving you a record that goes very far back in time, is the piston core. And this is the weight stack on the end of the piston coring device. You load tubes containing lead or concrete into the end of that. And then a long coring pipe is attached to that, which is steel on the outside, and you load it with plastic core liner that we can actually take out by extruding it through the weight stack, pulling out long sections of pipe and cutting them typically into 1.5 meter long tubes of mud.
On board this cruise, the crew will rig a 20-meter piston capable of sampling 12,000 years. Because launching the piston core can be a dangerous process, the ship's deck is cleared of all but the essential crew and scientists. The piston coring tool is winched over the side of the research ship just prior to deployment. When released, the piston freefalls 1,500 feet to the ocean's bottom. The weight causes the piston to impact the sea floor as it digs 20 meters into the sediment. Still connected by cable to the ship's winch, the piston core is carefully pulled back to the surface. Once safely on board, the piston is disassembled into shorter tubes ready for processing. The team works fast to package and seal the sections in order to preserve the unique qualities of the samples.
So, when we get back to the lab, when we split up the samples, we're going to be taking about 1-inch cubes into the side. Then these cubes are going to be tested for their magnetic properties.
Purge this with nitrogen now. Okay. So let's go ahead and do that.
We're hoping that we're able to preserve the anoxic state at the bottom of the basin. So we're going to be very carefully sectioning, bagging, and storing in a refrigerator all the pieces of the piston core. Once we cut it open, though, we're hoping to see the same kind of varve sediments that you get in the cast-in and box cores. And we're hoping to see a much longer record.
While the sediment teams continue on deck and in the labs, scientists interested in the ocean's food web prepare for their own work. Their plan is to launch the IKMT, a midwater trawl designed to collect large quantities of plankton as well as other sea life.
There are fish in the ocean that inhabit a region called the mesopelagic, or the middle layer of the ocean. So if we took a column out of the ocean and we said, well, the surface is at the top, well, the mesopelagic area would be 200 meters below the surface down to 1,000 meters. Now, to determine which ones are vertically migrating and which ones aren't, I'm going to tow a specific type of net at the surface, and then I will collect those samples and see what's in them, and then I will do the same net tow at a deeper depth. And obviously the ones that I get at the surface at night are vertically migrating. The ones that are at the deeper depths and not found at the surface are not vertically migrating. I'm interested to see where the transforming juveniles exist in the Santa Barbara Basin because it's a low oxygen area. And I have a feeling that the transformations will happen at different depths relative to other areas that are more oxygenated. Now, I can't use the IKMT to collect transforming juveniles and determine what layer that they came from in the water column because that net, when it's fishing, is open all the time. The mouth of the net is open. Now, the type of net I'll be using to collect the juveniles, or the larvae and transforming juveniles, is a system that opens and closes. So we have a system of nets, and it's a series of 9 nets that open and close. And the way that we do that, it's really fascinating. It's quite a large net. and a series of nets. And we sit here on the ship in the lab once it's been deployed, and we have a conductive cable over the side of the boat connected to the net that talks from the computer, and we tell the computer to tell the net when to close the net, so that I can say, oh, well, I know I'm at this depth, I'm at 500 meters. I'm going to open one and then close it at 400 meters. So now I know that whatever's collected in that net came from the layer of 500 to 400 meters. So if I do this a number of times in succession, then I can figure out where my animals came from. And if I can find the transforming juveniles, then I'll be really excited. And I'll compare it to other ones that I found in other areas and see if there's a difference based on the low oxygen compared to the oxygenated areas.
The team eagerly waits as the nets are pulled to the surface. The scientists help the ship's crew gather the nets and begin to process their catch.
She's gonna get the buckets, I think.
After the first deployment of the day, the crew and the scientists have worked out all the bugs and begin to collect the samples they've been looking for. As work continues throughout the ship, some scientists are looking for a little sleep, while others are just waking up for their work best done at night. Among those on the night shift, one team has an interest in the jumbo Humboldt squid.
Well, it's a very large, voracious squid that it can be bigger than me, actually about this size. And they are found all the way from San Diego to the mid part of Chile, approximately. But as you know, the ecosystem is changing. We have global warming. And since the 1997, we have that this squid particularly expanded its range. So it has been found even in Alaska and even the south part of Chile. So now that we know that the ecosystem is changing, we want to know how the species are also reacting to this. And in this case, what is the role of this species, Osidicus gigas, in the USA or the Canadian waters. These are the jigs. And as you can see, they are not nice. They are 2 rows full of big hooks. And as soon as the squid see these lights, these fluorescent lights, they go after, they get entangled with the tentacles and the arms, and there's no way to get out of the jig. So I recover the line and then I can have my squid.
Water sampling continues with a team of scientists interested in small things, microbes.
And this is our baby, the McLean pump. It's a self-contained battery-powered pump that we send down to a particular depth and pump water through the filter, which is in this housing up here, and out through this flow meter right here. So there's a little dial underneath there that tells you how much water flowed through. And we set the timer so that it's down there for about 4 hours and generally pump around 4,000 liters of water for every deployment. So we just retrieved it. So it's been down there for 4 hours, and we're going to offload the data that's contained in the little computer system inside the battery chamber. And then we're going to remove the filters so that they can be frozen in the -80-degree freezer and later analyzed in the lab. We're looking to extract the lipids from the microbes that we collect on the filters. So these are deep sea microbes from 475 as well as higher depths, so like 250 meters. And the end goal is to be able to compare the structures of the lipids that we extract from the microbes to see if there's any variations in the structure based on environmental conditions like anoxia, where there's no oxygen, versus an oxygenated region.
With equipment designed to capture, filter, and analyze water content at various depths, these scientists employ the CDT Rosette water sampler. This computer-operated water capturing device will open and close its tubes at different depths. In addition to recording conductivity, temperature, and depth, the Rosette is also equipped with a camera transmitting images back to the ship for observation.
And here, in the same frame, you can see an U-faucet, which probably you have heard about, is also known as krill. That's whale food. Basking sharks eat them as well. I've been taking samples of water, sample waters from the CTD Rosette from Niskin bottles, and as you saw earlier, probably I was preparing these samples to measure the oxygen, and now they're stored and they're going to be ready for analyzing back in land when we come back to land. So I have my samples ready, and the reason I'm looking for oxygen is because the species that I'm interested in actually diapause in this basin. It's actually a phenomena that these copepods undergo every year towards the fall, end of the summer, beginning of the fall, where they reduce their metabolism and create or build storage, lipid storages, and sink deep into the basin to be able to survive the winter conditions.
Back on deck, the shifts are changing as the night crew heads for the galley for a quick breakfast. In the next episode of Sea Science, follow the crew as research continues in the Santa Barbara Basin, and with new species being discovered in the most unusual places.